we're going to cover energy, and then we're going to cover genetic information. So energy is the capacity to do work, not like working at McDonald's, which I'm hoping that nobody does, or Domino's. Did you guys hear about Domino's Pizza? The program that they're using to calculate the pay of all the employees is faulty, and it calculates the hours on purpose. And they knew about it in 2011 or 12. I'm sure, and they knew about it and didn't do anything. So let's hope that energy is not the capacity to do work at Domino's. Note to self, never order from there again. Not that it tastes like cardboard. But light from the sun is the energy, a source of energy for all of our life. And if you don't use the light for energy like plants, then you're going to eat something that uses the energy from light, or you're going to eat something that eats something. So when you eat a cow, the cows eat the grass that uses the light from, okay. So we're non-photosynthetic. We should just consume carbohydrates, or we're going to consume animals that consume carbohydrates. All right, let's talk about this idea of the ball rolling down the hill. I'm sure you've seen before, and it's going to be something that's going to be in the back of your mind as we go through the rest of the semester. Talking about the energy of our reactions. When we talk about the reaction, we're going to do the what, but we're also going to do the why. And the energy behind those reactions is really going to be what is the why. And it's going to be something that we use to understand why these things are occurring. So any type of spontaneous reaction, the reactants are going to have more energy than the products. So if it's spontaneous, then it's going to, the products are going to have lower energy, lower intrinsic energy. Remember I said ATP is angry? All right, that means it has a lot of intrinsic energy. And ADP has less intrinsic energy. So that's what I'm talking about, the height of it. ATP would be up here, ADP will be down here. All right, so the products would have lower energy. So it's a spontaneous reaction for it to lose one phosphate. However, there's a little bit of energy that you have to put in order to get that reaction to go. What is that called? Does anybody know?
Activation energy.
Activation energy, right? We'll talk about that a lot later. But what your enzyme does is lowers that energy of activation so that it can go forward easier. And we only want that energy of activation lowered when we want the reaction to occur. So the idea is that the ATP has a lot more energy, intrinsic energy, than the products. Now, it doesn't mean that we can't go the other way. We just have to maybe couple a reaction. So you'll see this a lot. So say we have a process that the reactant starts up here and the product goes down here. It releases this much energy. Then we have another non-spontaneous reaction that maybe uses up this much energy. What you'll see is that we're going to couple a reaction that gives off a lot of energy to a reaction that uses up less energy. And if you put them together, you have a net loss of energy. So it's still spontaneous putting those two reactions together. You're just using that energy from the spontaneous reaction to drive the reaction that's non-spontaneous. So again, our redox reactions are those spontaneous energy-generating reactions. And that energy that I was talking about that's intrinsic, it's the energy of formation, or Gibbs free energy, G. And this delta, this triangle, means change. But it's very important that you understand that it's final minus initial. So the triangle is end minus beginning. I'm going to say that like 50 times because I think I went through my entire undergrad with no one telling me this. And I was always like, why is it negative? It's because, think about this. The ADP is down here. The ATP is up here. And so the ATP is up here and ADP is down here. So if final is a little number, an initial is a big number, then final minus initial is going to give you a negative value. Does everybody get that? A little number minus a big number, 1 minus 10, is going to give you negative 9. So that's why when you have the change in free energy being negative, it's spontaneous. If the free energy increases, you have the delta G being positive, it's not spontaneous. Doesn't mean that it's not going to happen, but you need energy to be put in. And that external source can be another reaction that gives off energy. When we pass electrons from one molecule to another, we're going to give off energy. Remember our sugar, and how I said we're going to take glucose from the beginning, start with glucose and bring it all the way down to carbon dioxide. We take, essentially when you have a fire in your backyard, you essentially do the same thing when you have a campfire in your backyard. It's so much fun. It's that season now, right? So you're going to burn it, and when you burn wood, you should get just carbon dioxide and water. So what you're doing, combustion, is oxidation. But you're doing it very rapidly. All that energy is given off as heat in the flame. What your body is doing is it's the same process. It's oxidizing the carbons on glucose. However, it's doing it in itty-bitty steps so that we can capture that energy. So instead of doing one reaction, the combustion of glucose, we're going to do it in many, many, many reactions over 3 different processes. And then we're going to, instead of capturing that energy in a flame, we're going to capture that energy in ATP. And so we're little batteries, actually. And actually, the electron transport chain can actually measure the voltage across the inner membrane if you had little bitty electrodes. That would be kind of... Redox reactions are definitely going to be the most important reactions that we're going to be studying this semester. And I call it the energy currency, or I think money molecule is more fun to call it. So again, if you see that a molecule has more hydrogens, it's going to be more reduced, and more oxygens is going to be more oxidized. So the more reduced the molecule is, the more energy it contains. Remember I was introducing fatty acids to you, and it had a lot of hydrogens in it. And glucose, every carbon had an oxygen already. Well, the goal is carbon dioxide. So if we're trying to oxidize up to carbon dioxide, fatty acids are basically at ground zero. Every carbon only has hydrogens or bonded to another carbon. But glucose is a step or two ahead, right? So if remember I'm saying we're getting to the same spot with both of them, carbon dioxide, but the fatty acids actually have to go through more steps, more reactions in order to get to carbon dioxide. The glucose doesn't have to go through as many steps. That's why you get more energy out of eating fat than you do out of carbohydrates. And so fats yield 9 calories per gram, while carbohydrates only yield 4 calories. The big C is for nutritional calories, which are different than our physical chemistry calories. So again, the transfer of electrons is not through a copper wire. Well, there is copper in your electron transport chain, but as hydrides, the hydrogen with an extra electron. So it's called a hydride unit. It's a reducing equivalent. And our holders of their hydrides are our coenzymes, the NADH. So NAD is our coenzyme that it's going to help oxidize or reduce things. So when it's NADH, it's holding that energy, and then it will give it off to something else. FAD is also another coenzyme, and we're going to study these in depth. I mentioned this before. Our pathways, our metabolism can either be breaking down or building up. And when you break down, it's catabolic. When you build up, it's anabolic. So when you are building up, you're going to a more reduced state. So you're going to, anabolic pathways are using energy, you're building, and so they're going to be more reductive in nature. where catabolic ones are going to be oxidative in nature. You're going to carbon dioxide and breaking down. So breaking down, oxidative, catabolic, building up, anabolic, reductive. Okay, lastly, we're going to talk about the information. What is making all of this go on? And I'm sure you've heard of the central dogma, where the information flows from DNA to RNA to proteins. All right? The structure of our proteins is what is better than the zeros and ones in computers. So here, this shape of this protein, particularly in the active site, is going to determine what it interacts with and be the information that's holding it. But proteins are made from RNA, and RNA is made from DNA. So really, what we're going to do is in the nucleus, we're going to transcribe, or in transcription, make the RNA. The way I like to think of it is when you transcribe, the definition of the word transcribe is to write over, is to copy. So in the same language. So you're using nucleotides from DNA to nucleotides in RNA. You're just copying the information over. But in translation, you're translating from one language to another. So you're going from nucleotides to amino acids. So that's the way I remember the difference between transcription and translation. Transcription, you're transcribing, just writing over the same language, nucleotide to nucleotide, DNA to RNA. And going from RNA to the proteins is translation, because we're going from nucleotides to amino acids. So RNA is made on that DNA template. In translation, the protein is synthesized from the 3 types of RNA. All right, so transcription, what's happening? I have a nice little video at the end of the semester that we're going to watch. But inside the nucleus in RNA polymerase actually runs down the DNA, unzips it, and there's an intake sort of pore where our RNA nucleotides come in, and then you're making a copy, complementary copy of messenger RNA. This messenger RNA then leaves the nucleus, I don't care about that, then goes to the ribosomes, and in the ribosomes, this is so stinking cool. In the ribosomes, you see that it lines up. So the messenger RNA comes in and goes into 2 different spots. And this transfer RNA on one end has the anticodon, which matches up with 3 base pairs in the messenger RNA, which has the codons. And then on the other end, it has an amino acid. So the blue little ball is the amino acid. So this funky-looking transfer RNA comes in to the ribosome and then lines up the anticodon to the codon, and it has an amino acid. Another one will come in to the next stop. And then what happens is the ribosome catalyzes the formation of the peptide bond between the 2 adjacent amino acids. Put it all together, because I want to go play tennis, although I'm really bad at it. So here you start out with our atoms, you make your biomolecules, then you can make your organelles, your cells, tissues, organs that make up our person that's going to go use their ATP to play tennis.